Good. So let's slowly but surely start our webinar. 1 second, I will show my screen. Good. So welcome to everybody. We are very happy to continue our series of webinars. This time we will speak about the drought and stop stress program to deal with different stresses, not only the drought but also some others, even of anthropogenic origin, as you know, some problems are caused by the weather, sometimes also by our actions. So again, we will have a recording of our webinar available to you and to our partners later. So if you cannot listen to the webinar, you will have the opportunity. So for those who join to our content first time, just a few words about ourselves. So we are BTU group of companies with more than 500 people. We have more than 60 biologicals in portfolio and apply them on more than 4 million hectares each year. And we have a big database of trials, more than 2,300. In fact, it's almost 3,000 at the moment. We are updating this information for next week conference in Madrid. And right. So we are based in Ukraine, but also have office in Germany since 2019, and already 25 exporting to 25 countries in the world, starting from Ecuador and up to Japan, but mainly in Central and East Europe. We have more than 20 products registered in different European countries. So if you are from Europe or around, it's quite easy to make registrations of our products or mutual recognitions based on these European registrations. This is how our factory looks like in west part of Ukraine. We have quite a huge capacities with fermenters up to 60 cubic meters and all the special equipment and technologies to provide clearness and effectiveness of all products of each liter of the products. This is how our office looks like in Kiev and in Germany and parts of our team. And as you probably know, we are quite active in the industry, so we know what is happening and what are the latest trends, and we are quite often among the opinion leaders at these events, sharing our expertise with the industry. By the way, next week you can find us in a New IG conference in Spain. For those who are not yet with us, this is how we work with our partners normally. So starting with selection of the products, registrations, patenting, education, certification, trials. We are close to you, helping to and sharing our knowledge and experience to find the best solutions for your conditions and to help you to find out its efficacy. Also, we organize in meetings of our international partners annually. This year it will be in June in Germany. So we will be happy to see you face to face on this event. And let's come closer to the topic. So we will speak about stresses, but also it's important to understand what is happening at the moment on the market. So we are quite actively communicating with partners and farmers in different parts of the world. And now, because of the war in Iran, there are some disruptions of supplies of energy sources, and production of fertilizers, especially nitrogen fertilizers, again have some difficulties, and that is immediately can be seen on the prices of those fertilizers. So if you see, it's not the big prices up to $1,000 per ton as it was in 2022, but still it's already quite a big increase of the price, somewhere around 50 to 60%. Both on the examples of urea and the ammonium phosphate. you can see that now prices are quite expensive, but I believe you have this information from your agricultural reality as well. But what is even more, not even more important, but as important as this, that there is some problems with fertilizers availability. Just recent week, for example, I spoke with several farmers in the Czech Republic, in Poland, that it's difficult to find one on the market, or the urea or other nitrogen fertilizers, because of the change of prices, etc. So we we expect that this season farmers could face some challenges in nitrogen fertilizers supplies, and that is why we believe that one of the stresses which plants could have this season could be lack of nitrogen nutrition. And this in this direction, we also have some solutions, so we will show it to you later today. And here I also put the indication of prices of grains of corn. So you can see again, more or less for last recent 2-3 years, it's quite stable. So again, we understand that for farmers now it's quite difficult uh, to have intensive technology, because they need to spend like 50% more on the nitrogen, while they have the same price of the commodities. And we don't know where will be the price, but probably it can remain on the same level, because we see on the 10 years that it's more or less normal price for the grains. So we understand again the difficulties for the farmers, and we try to provide some solutions how they can optimize their cost of technology. And one of the again popular or widespread topic about stresses, abiotic stresses, is the drought. So you can see again there are a lot of talks around the drought now in agricultural industry. And here I find some interesting visualization from the resource you can see here about the risks of drought in agriculture in different areas. So again, in Europe, it's still quite kind of safe in Central Europe, but if we speak about Spain, Balkans, and East Europe, Asia, then Middle East and North Africa, South Africa, and US, these risks are quite significant. And for some main crops like maize, you can see that the risks of losses of the yield because of the drought are quite significant, even from the moderate drought. That is why, again, we believe that drought is quite a big, insignificant risk to agriculture, both in Europe but in other regions of the world. And that is why recent 5-10 years we invest a lot into the studies how we can help plants to continue growth and save productivity in such drought conditions. So on this, I would like to give a word to my colleague, Yaroslava Buchonska, who will tell us more about kind of science and mechanisms of action of biostimulants, how they can help plants to grow in these stressful conditions.
Hi everyone, do you hear me well? Could you please tell me?
Yeah, we hear you.
Great. So let's start now. My name is Yaroslava, I'm a plant physiologist, and I'm happy to see you all coming to get some maybe new information for you. I hope it will be useful for you about how we can help our plants to overcome stressful conditions with the help of natural microbial antistressants. But first of all, I would like to start with some biological facts about how actually plants are living under stress, what's going on in their organisms. So basically, even though we know there are different kinds of abiotic stresses. On this diagram, I am talking only about abiotic stresses, but we have to keep in mind that we also have biotic stresses, those which are caused by different pathogens. So abiotic stresses, they combine ultraviolet exposure, drought, high temperatures, low temperatures, hail, frost, mechanical damage, salinity, unoptimal pH, and heavy metals. But even though these inputs are extremely different, plant responses are pretty the same to all kinds of stresses. First of all, plants are reacting with decreased photosynthetic activity because they are mainly closing their stomata, and stomata are working normally for transpiration, and when transpiration is off, carbon dioxide supply is also off. That's why photosynthesis is stopped. And this also triggers generation of reactive oxygen species. Reactive oxygen species are, on the one hand, very important messengers to inform all parts of the plant that something bad is going on. They are used for signaling. But when they are produced in excessive amounts, they are really harmful because they are damaging everything they meet on their way, including all crucial biopolymers like DNA, proteins, cell membranes, everything they meet. That's why they are very harmful. And all these effects, they cause decrease in plant biomass accumulation, they cause decreased plant growth, and of course they are decreasing yield, what is the most interesting for us. And this scientific picture is describing the role of plant growth promoting rhizobacteria in abiotic stress resistance of plants. They have developed different mechanisms which are helping plants to overcome negative events, which you can see on this picture. And there are some mechanisms using which these microbes are making this anti-stress effect. So let's take a look. So they can produce different phytohormones, including, for example, cytokinin, which is crucial for plant growth and development. They are also triggering production of antioxidants, which are very important to combat with reactive oxygen species and oxidative stress. And very, very interesting mechanism. They can decrease excess ethylene production. Ethylene is a stress hormone, but when we have too much ethylene, plants can abort flowers and leaves. They are highly stressed, and a unique feature of plant growth promoting bacteria is that they can produce ACC deaminase. It's a very specific enzyme that degrades ethylene precursor, and with the help of this enzyme, ethylene production is lowered and plants are less stressed, we can say. And we can also see there are different other phytohormones and biologically active substances which are triggering alteration of root morphology and different adaptation reactions in plant. And this all helps plant to maintain their metabolism and growth processes. And here is what happens on the cellular level when abiotic stress is coming. You can also see that despite that we have different stresses, reactions inside cells of plants are pretty consistent. They are all causing different biochemical and morphological changes. And as you can see, everything is connected to calcium signaling. Without calcium, there is no proper messaging across plants. So here is actually one very important role of calcium. Don't forget about this nutrient in your fertilization systems, because it's very important messenger in plants. And actually all stresses, what are they doing? They are finally triggering changes in gene expression. So gene expression is how genes are working. So we have nucleus here, we have some chromosomes, we have DNA, and the way genes are working is called gene expression. And these events, they can change the way it works. And after that, plants are changing their morphology, they are growing more roots, they are accumulating some substances. So everything is due to genes work. And on this scheme, I want to summarize some negative effects on the left side of this picture, which are caused by different stresses, and on the right side you can see some positive effects which are triggered by beneficial microbes. Beneficial microbes, also known as plant growth promoting rhizobacteria or fungi, they are known to increase photosynthetic rate of plants under stress. They are also known to improve osmoregulation. So osmoregulation is the ability of plant tissues to maintain water balance, to have enough water to run all metabolic processes. And they're also enhancing root development, and they help plants to mitigate oxidative stress. And with all these effects combined, we have plants which are more resilient to different negative natural events. And there is one very, very interesting mechanism because of due to which plants are better adapted to stress. So it is called priming. And it is actually the way how microbes are kind of teaching plants how to survive better. So a few words about priming. Now we know that plants have kind of stress memory. So when a plant is encountering its first stress in its lifetime, before any stress, this plant is called naive plant. And after any kind of stress, plant is developing some adaptation reactions. And if they are successful, so if these plants actually survive, because outcomes may be different. In case of survival, this plant uses its kind of memory to memorize these reactions to be more efficient after stress event. And this memorization is called priming. But what is more interesting is that it can be caused, so this priming state can be caused not only by different stresses of different nature, but also it can be triggered by communication with beneficial microbes. So microbes are producing some specific biologically active compounds which are triggering plant defenses, and they are making them more robust, and they are changing their response to stress, and they can react more faster and more intensive to stress. And what is also very interesting is that they are becoming more sensitive, so they are reacting from a kind of lower threshold, we can say so. It's a very interesting phenomenon, and it is now widely studied by different scientists, and we still have to understand how actually it is activated, what is the basis of this phenomenon, but it's very interesting that we can use it in our practice. And let's move on to some of our products which contain these beneficial bacteria which can trigger these reactions in plant. And I would like to start with our very well-known product, Azoto, which is known for plant growth promotion and nutrition. It contains free-living nitrogen-fixing bacteria, which is called Agrobacterium puissans. It is able to fix nitrogen without forming any nodules, like as we know for legumes. It doesn't form them, it fixes nitrogen just as it is. But when we apply it in a foliar way, it also works as a very nice biostimulant because it contains a lot of biologically active substances which are triggering immune processes in plants and making them more strong. Our second product I would like to tell you about is our liposome. It's bioadhesive, and it is made of polysaccharides of microbial origin. And you can remember a lot of different very interesting facts about liposome from our previous webinar. But this product is also interesting for us in context of stress mitigation, because it not only protects plant during their vegetation period after application. These polysaccharides are also working as kind of messengers because they can tell the plants that bacteria are drying, that supposed bacteria are drying, and bacteria nearby plants are stressed, and it means that plants have to also activate their protective mechanisms, because if bacteria nearby are stressed, then it means that stress are coming and they have to be prepared. And the third product I would like to tell you now is organic balance. It is our biostimulant, which is made of complex of bacteria which increase plants' resistance to stress factors due to their active metabolites, and they also stimulate plant growth and development, and they improve plants' nutrition. And a few words about our field experiment, which took us 3 years in northeast region of Ukraine. This experiment was conducted with the use of maize, and we have recorded limited amount of precipitation during critical growth stages, and plants in this experiment were treated foliarly with biologicals in a very early stage, because it's very important. I didn't mention it when I was telling you about priming, but it's very important to know that this priming effect can be activated in plants when they are growing under optimal conditions, or under conditions which are close to optimal, because plants need, first of all, some time for these priming reactions, and secondly, they need some resources to rebuild their metabolism and plant architecture. So it takes time and resources, and that's why it's good to do it before stress events than after. So in our experiment, plants were treated during very early 3-5 leaf stage. And what we analyzed, of course, we analyzed yield, average yield for 3 years, but we were interested not only in yield results, but also we wanted to know which processes in plants led them to be more efficient after biostimulant treatment. So we have also analyzed relative water content in leaves, antioxidant activity of them, because oxidative stress is very severe during any stress event. And also we were interested in relative gene expression analysis. We have selected 3 marker drought stress associated genes So these three genes were shown by other scientists in their experiments that when plants are heavily stressed with drought, these genes are working extremely actively. That's why we selected them as markers. Let's move to results. As you can see, with the yield results, this biological treatments, they resulted in better yield accumulation in plants compared to control. This is average yield per 3 years, so you can see pretty consistent results because plants which are working more efficient under stress, they have, of course, better productivity. Secondly, we analyzed relative water content in maize leaves, and you can also see that plants which were treated with biologicals, they have better water high water content in their leaves, and water content is crucial for all metabolic and biochemical processes. It is crucial for photosynthesis, for growth, for all growth processes, and that's why we are not surprised that these plants actually have higher yields. And the third, the most interesting to me as a biologist, a relative gene expression in leaves. So you can see we took control plants gene expression as 1, and we compare all treated plants to control, and we can see that all treated plants have actually a lower gene expression of drought stress marker genes, and we can assume that under the same conditions which these plants in which these plants were growing, some of them were more stressed, like control plants, and some of them, like treated, they were less stressed, and that's why we have better yield results. And the last but not the least, we have also analyzed the antioxidant activity in maize leaves, and for some of them, antioxidant activity of leaves was also elevated after treatment with some biologicals. And it can also explain some effects because oxidative stress is very damaging to plants. And when we can lower even somehow antioxidant oxidative stress, we can have beneficial results. So to conclude, you can see that in our experiment, plants which were treated with biological, especially with a mixture of organic balance, azotohelp and liposome, these plants have the highest relative water content, and actually the treatment with biologicals led to a reduction of the expression of stress-related marker genes, especially, drought stress marker genes. And we also have noticed a significant increase in crop yield, which is the most interesting effect to farmers. That means that biologicals can be used as a part of sustainable agricultural strategy, especially in our very different and very difficult climate conditions, which are dramatically changing with globally on our planet, and that this strategy to reduce synthetic pesticide use can also help our environment. So I hope this information was useful for you, and you can also use it on your plots, on your crops. And thank you for your attention.
Thank you very much, Yaroslava. Very insightful and interesting as always. We have received one question about the registration of our biostimulants on a European level. So I can comment briefly that we speak now about 3 products, AzotoHelp, Organic Balance, and Liposome. So AzotoHelp is a nitrogen fixing product. It is registered on the national level in different European countries, and via mutual recognition, we are bringing it to other countries. A similar story is about Organic Balance. And speaking about Liposome, it's registered as an adjuvant. also national level in Germany, and we are doing mutual recognitions to different countries. We are observing changes in European regulation, and we are looking forward to the updates of the positive list of microbes, and then we will consider to register some of our solutions on the European level as well. But if you have any insights or proposals for us, feel free to contact us via email, and we will be happy to continue the conversation. Before I give a word to my colleagues to share some practical experience, I just want to show some kind of summary slide about types of stresses. So again, Yaroslava mentioned it. We have some abiotic stresses, but we almost don't speak about them because that is more about plant protection. But then there are abiotic stresses, and among them climatic conditions. So again, we mainly speak about temperature and precipitation. Also, we are studying liposome now for the sunburns protection, so it's a kind of light stress. Then we have a lot of practical examples of anthropogenic stresses. For example, pesticides exposure, soil cultivation, fertilization system, and my colleague will show some of them. And also we have some examples of physical and chemical properties of soil, which can influence and make stress for the plants, and we can help them to grow in these stressful conditions. And also a very small comment that normally we have three alternatives how to apply biostimulants before the stresses to help plant to be more resilient to the stress, at the same time to reduce the stress, and also after exposure, because very often after the stress plant is kind of preserved and don't grow, and it needs time and resources to recover the stress. So our idea is to help it to recover growth after stress faster and not to lose productive time of the season. So on this, I would like to give a word to my colleague. Dmitry, could you please share with us our practical experience with field crops of our program?
Yeah, of course. Hello to everyone. Do you hear me well?
Yes, we hear you.
Yeah, nice. So, now we discuss about our field practice with this program. Innovative program, like my colleagues already mentioned, we will discuss about organic balance, azoto health, liposome in one tank mix in different types before, after, or in period of stress. We will recommend to apply this complex of products. I would like to share this table for you. As you see, we have small statistic with different years. It means that in different years was a lot of different climate condition. Also, as you see, we used this program on the different crops. And also we used in period of some extremal change in the temperatures after frost stress, after pesticides load. Also we can use this program, stop stress program, together in one tank mix with some herbicides. Also, I would like to draw your attention that we can have a lot of different stress on the plants after a lot of chemical products like foliar herbicide, and also in few countries last few years very big problem was like stress after fooding. As you see, for different crops, for different stress factors, after application of our innovative program, Stop Stress, we can help to recovering or to help plants to recovering after stress, and also in the same time we can provide the increase in the yield. A little more details about some trials. As you see, we used our program on the winter wheat. We used a little bigger rates of each product in this program because, as you see, we had two stress like after chemical products and also after frost stress. So in this case, this program with bigger quantities for each product provided us increase in the yield plus 0.6 ton per hectare on the winter wheat. One more trial with this program. What stress we have? We have stress on the plants after effect of soil herbicide. It was on the sunflower. On picture from right side, you can see what plants, how plants looks before application of our program stress, stop stress program, and after application. So application once of this program provided us increase in the yield plus 0.4 ton per hectare increase in the yield. And also you can see on this picture what different we had in vegetation period. Left side it is control, right side it is after application. One more stress after stress situation where we had a herbicide leaching. As you see on this picture, first picture before application, we see that our plants like after some burns, and for right photo it is 45 days after application. It is also was sunflower. We used a standard scheme of our program, and also one application of this program in phase 2-3 leaves provided us increase in the yield plus 0.7 tons increase in the yield for sunflower. One more on sunflower. As you see, we have a lot of trials on this crop because farmers used a lot of some herbicides. And in this case, as you see, we make the some combination in tank mix to our program plus Eurolighting. Eurolighting, it is like foliar herbicide. And also, as you see, we use this program in period in phase of development phase of crop for leaves. And this combination in one tank mix provided us plus 0.3 tons increase in the yield per hectare. Also, we have a trial on the corn. On this slide and with this trial, I would like to draw your attention that we used this program in period of 7-8 leaves of plants. And also, as you see on the control base was very high yield. Even in this case, we provided increase in the yield plus 0.3 ton per hectare. and some new trial, as you see also on the form. We use the in west part of Ukraine standard program like organic balance half a liter, azoto help 0.3 liters per hectare, liposam 0.3. And also in this case, our program provides us increase in the yield around 0.26 tons per hectare. A little more practice with the products. I would like to draw your attention because we have some change in the program. In this case, we use the Azoto Help dry formulation, and the rate was 150 grams per hectare, plus liposam 0.3 liters per hectare. On this trial, as you see, was very high temperature, and crop was sorghum, and one implication of I can say like half a program provided us increase in the yield plus 0.42 tons per hectare. You can see some different in development of root system, and also you can see better development of vegetative mass. One more trial, as you see, we checked a few types, few products, like for preserving cultivation, we use the liposam with soil herbicide. It is our recommendation for better fixation of and prolongation effect of herbicide, and also our stop stress program. Very interesting that it was on sugar beet, and this complex of products, complex of applications, as you see, provided us increase in content of sugar. As you see on the control was 18.4, on the trial was 18.8% of sugar. And also in the in general on the control was 80.6 ton per hectare yield, and on the trial place was 103.6 ton of yield. And also on the picture you can see development of plants from control side and from trial side. One more trial when we had a competition with some competitors' product. As you see, we used only Azoto Help from control. Yes, we have like native place. It is control. Our trial where we use the Azoto Help half a liter in the stage of 7-8 leaves of corn, and the competitive product we used with this amount like 0.333 kilograms per hectare in the same stage like our products. I would like to draw your attention on 3-4 last columns. These columns told us that, as you see on the Azoto Help, for example, in period of 10-12 leaves, coefficient of photosynthetic efficiency, photosynthetic work of photosynthetic apparatus was higher, the same in flowering period than our competitor product, and also quite better in comparison with control place. Also, one more interesting trial where we use the phytobact. It is product which based on Bacillus subtilis. We use this product like to see what amount of water plants use in period of usage of our product. So as you see, I would like to draw your attention where we have a red color. On this, in these results, you can compare yield yield of crop after application of phytobact in comparison with control. As you see, with phytobact we had a 8.67 ton per hectare, and on the control place was 8.48. In the same time, as you see water use ratio, we see that after application of phytobact, plants formated bigger yield but used less amount of water in comparison with the control phase. Yeah. So from my part is all.
Thank you, Dima, a lot for these practical insights on most field crops. So on this, I would like to invite our next colleague, Oleg, who will show us examples of how you can use biologicals to deal with expensive nitrogen fertilizers. We have plenty of such examples. But while Oleg is opening presentation in full screen, we have some small questions here. First, what is the survival rate of bacteria when dried or coated? Indeed, for most of our key products like Azoto Help and Organic Balance, we have a dried formulation in WP formulation, and we work a lot to optimize this formulation to have sufficient level of bacteria in them. So I cannot tell exact figures because again, if we speak about the declared volumes, they are not less than in liquid formulations because we also concentrate bacteria before drying. But again, it's more technological questions. The main idea is that products are available in liquid and dry forms, and in dry forms they are not less sufficient in terms of the titer. So Oleg, please continue with the presentation. Could you open it to full screen?
Just a second.
Okay.
Just a second. I will.
We are glad to see you in full screen. Also waiting for the presentation. While you are running the presentation, I'm very happy to see new faces here, and also glad that people even from Colombia join us. So we are glad that we choose the time suitable, even though in Ukraine it could be already a bit tired because it's end of the working day, but we managed to have here some people from other parts of the world. I can start presentation on my screen. So give me a second.
I think it will be better.
So okay, we can start.
Okay. My name is Oleg Baranovsky. I'm agronomist at BTU, and in this small block I will show you how we can manage another stress of farmers. This season is stress with fertilizer cost and some troubles with deliveries of fertilizers. So in this case, it's a decrease of fertilizer rates with 20%, and how we can substitute it with our products. On this example with potato, we applied ground fix, our PK mobilizer, Azoto help like nitrogen fixing bacteria, and mycorrhizal product MycoFriend to substitute 20% of NPK. And in this case, we also achieved increase in yield, both in full fertilizer rate and also in decrease. So in this case, we can show that we can not only substitute fertilizer, but also improve efficiency of their use. So next one. In this 2-year trial in Ukraine, we investigate how use of Azoto help with decreased rate of fertilizers in different ways of application. So in this case, decrease by 30 kilograms per hectare of nitrogen fertilizers on corn. We see the good result as under cultivation application with liquid formulation and also with powder one. Also, we have good result with foliar application in 3-5 leaf stage, and combination of this product under cultivation and foliar application too. In all of these cases, we have good yield increase, and it was even better than full fertilizer rate in every of these cases.
Like, I'm sorry, did you mention also that it's 2 years results?
Yes,
average?
of course. Yeah. So another trial of substitution of nitrogen fertilizer, it's 3-year trial in Czech Republic. Here was substituted 160 kilogram of urea. On control plot, it was 200 kilograms of urea, and on trial, only 40 kilogram, where was applied Azoto help. We have two different variants with half of liter with Azoto help and splitted application of 0.25 liters of Azoto help in the different growing stage. And both of this application, we received good increase in green mass of corn and also in dry matter. Also, one of the case where we can substitute fertilizer, it's starter fertilizer. In this trial, we substitute a liquid complex fertilizer during sowing of corn, application of dry formulation of Azoto help with seeds in seeder show even better results than in further application of liquid complex fertilizers. And also combination of Azoto help and liquid fertilizers also showed even better results. So as we mentioned before, we can improve fertilizer use with our products. And in this also the same condition, 2 years trial on sunflower, but in this case we only compare substitution of liquid complex fertilizer with Azoto help and see that it almost the same result we have compared to usual fertilizer with our biological products. And another one example from Ivory Coast. Here was applied complex of our products. It's ground fig, Azoto help, and liposome, also with full rate of fertilizer and 25% decrease rate of fertilizer. In each case, we received good results of yield increase. I think compared to 100%, it also good numbers. And one of the good point for our product liposome, that application only of one of this product show how liposome can help resist drought condition and improve drought tolerance in this condition of Ivory Coast.
So thank you, Oleg, for this interesting experience. Again, we work with this direction of substitution of fertilizers already around almost 10 years and have a big amount of data, including nitrogen fertilizers and other types of complex fertilizers. So it is possible. In our standard recommendations, it's around like 20-30% of fertilizers. Normally, fertilizers cost more than the products you substitute them with, but you also can have an upside. And again, we assume that this year, in conditions of expensive nitrogen fertilizers, you have a nice chance to improve your economic efficiency of technology. So with this, I would like to continue with some other examples of components of stop stress program on different vegetable and horticultural crops. So first of all, example on the beetroot, you can see that with application of a few biologicals, Abiotic for seed treatment, ground fix for phosphorus solubilization for preserving cultivation, but also organic balance as a main biostimulant in our line during vegetation. We get quite sufficient increase of the yield of beetroot. It can be compared to the after effect of the manure applied on the previous year, but also you can see that biologicals help to kind of improve uptake of the nutrition from organic fertilizers quite efficiently. Also, you can see that we have quite positive influence here on the quality parameters as well. So another experiment on the cauliflower, also quite sufficient improvement of the yield. We tested different concentrations and or dosages of organic balance, in this case from 1 to 3 liters per hectare. So each of them was efficient, but with 3 liters per hectare, it was the biggest increase of like up to 5 tons per hectare of the yield, and also improvement of biometric parameters of this plant. Another example on Napa cabbage. Also, we tested organic balance, one application together with liposome in comparison to also other scheme with MicoHelp, our another biological product. And in both cases, we received around 2 tons of extra yield of this crop. So cabbage is quite sensitive to biostimulants, and organic balance together with liposome are quite good way to improve their productivity. Then quite an interesting study we did in Bulgaria. It was done by Center of Plant System Biology and Biotechnology. They tested liposome on cucumbers in controlled conditions, and where they reduce water rate 2 times. And we can see that use of liposome foliarly helped plants to grow better in these conditions and to keep the productivity on the good on the better level. And not a horticultural trial, but a trial in control conditions. Actually, we find out around 5 years ago that liposome have these nice properties to improve drought tolerance of plants. And we have plenty of studies in these directions, some of which Yaroslava showed you. But also last year we repeat the study to make drought conditions for small plants. And especially when there is a repeated drought stress, plants survive. When plants are treated by liposome, they survive much better. So this test is quite visual, and for what liposome can do to help plants to survive in these conditions. Then we used the full program of stop stress also in onions on different fertilization kind of backgrounds. So if we take just clean background, the effect of stop stress program was around 400 kilograms per hectare. When there was a good, better fertilization background, we see that the impact was quite significant, both for the yield, but also for sugar content and vitamin C content in the onions. And with organic fertilization, with manure, also we reach quite good increase of the yield, around 10%, and also improvement of sugar and vitamin C content. These are the photos of this study, so even visually we could see the better development of the onions, and the program was applied in BBCH 12. Besides that, we have some studies in orchards. This study was done in conditions of apricot orchard, and when we were preparing this slide, I was joking that it's a resistance to frost and hail, but it's a kind of resistance to health, because really the first was a frost, and after some period of time there was also very big hail conditions, and even in these conditions we succeed to preserve the crops and to have the fruit. Another study was some period time ago in Serbia. We tested organic balance together with phytobact and also ground fix in fertigation on the pears. And during the experiment, there was a heavy rain, and the orchard was flooded, which influenced the productivity. And it was quite interesting to see that the use of biologicals helped pears to survive and be productive in these conditions. And the increase of the yield was around 30% from hectare. And another study in Serbia was on raspberry. There was frost, and plants were quite kind of damaged. And you can see that comparing control plots and trial treated with organic balance after the frost was looking much better and greener. And not besides those key products, we have some other examples also connected with stress for plants. So we had an experiment in Spain around Valencia on citrus orchards, and we measured soil enzymatic activeness of soil and also NDVI indexes. And we can see by control indicators, then during May, June, it was quite hot, and the plants begin to lose kind of photosynthesis activeness in the fields. But where we used Ecostern in fertigation, we succeed to keep the photosynthesis still active, and also we succeed to keep the enzymatic activeness of soil with a better on a better level. And another stress of the crops is especially like in Portugal and maybe Mediterranean, so some on the grapes or cherries. Sometimes there is a big change of temperatures during the day, also a big amount of moisture during ripening period. And in this period, berries tend to crack. And when we apply liposome, we can preserve up to like sometimes 50%, sometimes a bit less, but still to have a positive influence on preservation of plants. Preservation of berries during these dangerous periods. And as a kind of summary, again, we have plenty of examples when this scheme helped us to preserve plants in orchards and in field crops with different stresses, anthropogenic, abiotic, like again, use of some pesticides or frosts or hail, etc. So we have different approaches to apply this complex before the stress, so that we can help plants to be stronger at the time of stress. We also can apply it together with stressful factors, so that again we mitigate the effect of the stress and plant kind of damage from it. And when we apply this complex after stress, we help plant to return growing processes faster and not lose important productive time. This year we were quite happy that our this program was kind of highlighted on Agra exhibition in Bulgaria. With our partners on Amitica, we receive a diploma which highlight award of this program. In Bulgaria is among the regions which are especially sensitive to drought and other abiotic stresses. So we already have plenty of experiences there where this program helped farmers to keep their plants alive and productive. So again, we are very happy with this award, but for us it's very important the impact of this technology to the agriculture, and we want to be helpful with this program to other partners, farmers, and regions. So on that, it's end of informational stage of our webinar, but we have prepared as the last time some questionnaire, small questionnaire for you where you can win some presents. And before we go into it, I will just quickly look through the questions which we have. So we also have some question about Kazakhstan, and yeah, so we will contact with Mr. Alexander about this topic. But what is important, we have more than 5 years of experience in Kazakhstan and this region, especially with Stop Stress program, and in conditions of big drought and lack of moisture, really this program helps to plants be more productive. So on this, I give a word to my colleagues who will start this questionnaire. Please use your telephone to scan this QR code to participate in the program, and with this you can win some useful presents from us. I already registered, did you? We see here Johanna. So please take a chance. Also, we will announce the winners after this small questionnaire, but you will be able also in Europe to win some samples of this program to be tested in your facilities. And for others, we also have some valuable gifts. So how many people we have already ready for the test?
4.
4 people. So I think let's start. For those who don't join, you can write your answers in the chat. So the first question is, which enzymes produced by PGPR bacteria help reduce ethylene levels in plants and improve stress tolerance? Catalase, amylase, ACC, deaminase, or peroxidase? Quite not an easy question, I should say. Let's see how many right answers we will have. So Yaroslava's, it's probably from Yaroslava's presentation.
Yes.
Yeah. So we have one correct answer, right? Good. Let's continue. What does the priming effect mean in plants? Reduced growth, loss of moisture, faster and stronger response to stress, or suppressed photosynthesis? I believe we should get more right answers in this question. Okay, let's try the next one. Which product forms a protective elastic layer and helps return moisture on plant surface? Azoto, ground fix, organic balance, or liposome? That one also should not be too difficult. Yeah, so indeed we have a correct answer, and liposome, due to polysaccharides in this composition, helps to do this elastic protective shield. Next question is, which indicator increased in maize after treatment with biological product? Soil acidity, heavy metal content, relative water content in leaves, or leaf temperature? So what we can preserve in leaves is our products. That is relative water content in the leaves. The next question is, what is Azoto Help mainly used for in practice? And we have here weed control, atmospheric nitrogen fixation and growth stimulation, soil acidification, and yield reduction. So seems like also Azoto Help is not a strong herbicide, but we can expect something from it. And that is atmospheric nitrogen fixation and growth stimulation. And we have next question. What is the main practical benefit of the Stop Stress program? Reduced seed quantity, lower soil moisture, reduced photosynthesis, or improved drought tolerance and higher yields. So I believe this one is also quite obvious. And we have the correct answer. So it seems like all the presents will go to our leader in our questionnaire. So another question is, what is one benefit of using biologicals in sustainable agriculture? Reduce environmental stress impact, more synthetic pesticides use, lower seed quality, or faster weed growth. And we have the right answer for better growth of the crops. And the last question, in which crop was the drought tolerance improved with liposome in Bulgaria? Soybean, potato, cucumber, or the wheat? Who was the most attentive during my presentation probably have seen the answer. So we have here the answer that it was cucumber. So yeah, we have the winner here, who is the Dimit. Please, Dimit participant, contact us after the event, and we would be happy to present you your small gift. So again, on this, I would like to thank for all the participants. We understand that agricultural season is going on, and most of you are already busy in fields, and it's difficult to have more time in this period of time. But still, we gathered around 30 participants from 14 countries, from different parts of the world, including Bulgaria, Nigeria, Spain, Egypt, Latvia, Finland, Czech Republic, Sudan, Austria, Germany, Ukraine, Greece, Colombia, and Cyprus. If I missed you, sorry, but we were very happy for everybody who joined us today. Oh, by the way, I see also probably Algeria here. So at least 15. So again, thank you very much for joining us. Soon we will share with you the presentation and the recording. Stay with us, follow our news. We have a lot of useful materials available on our website and our YouTube channel. Most recent presentations from industrial events, like the recent one about EcoStern for improvement of for acceleration of the composition of plant residues and improvement of soil properties. And next week, join us in Madrid, and we will present our update for our big database of trials. Now we have more than 2,800 trials already in it, but still we are calculating the final figures. So see you during next events.